the opportunity to talk about our research. So I'm going to present to you our recent deep learning-based method that extracts features from a collection of different types of protein-protein interaction networks, and then it uses these features to predict function. So this is the outline of my talk. So since this is a network-based method, I'm going to try to give you just a brief one-slide summary of network-based methods for function prediction, including some very old methods but very good like GeneMania, but also some very recent methods like mashup that was recently proposed. Then I'm going to switch more to and focus more on network embedding methods that start from a network and then they learn low-dimensional features representing nodes in the network and preserving the structures, their structure. And then I'm going to present to you our method, which is a type of network embedding method, which we call deep network fusion or deep NF. And then I'm going to show you some of the performance results of our method applied on networks downloaded from StringDB. And if I have time, I'm just going to mention some of our future work. So the major assumptions that we make when building network-based methods is that function is often shared between proteins that physically interact, or that have similar topological roles in interaction networks, or that are part of the same complex or a pathway. So therefore, we could say that networks are very good source of information for inferring function. On the other side, thanks to the advances in like experimental techniques that enabled construction of different types of networks, we now have different data like networks sources that we can use for basically predicting function. So we can now construct, let's say, physical protein-protein interaction networks where nodes represent proteins and the physical binding is represented as links. Or we have also genetic interaction network where nodes are genes, and then deviation from the expected phenotype under the various combinations of mutations of these genes is what is used to construct a link between genes. Then we also have gene co-expression network, which is just a correlation between expression profiles constructed for genes over different conditions. So basically, all these networks, they represent different and often complementary sources of information that we can use for protein function prediction. And one of the challenges is to develop network integration methods that can combine all these networks and predict function. So the setup for these network integration methods is that you have a collection or a set of networks that are constructed over the same set of nodes, but they have different wiring patterns or different connections that kind of carry different information. And the methods that can be used for function prediction either integrate these networks into a single integrated network, and then they use some sort of some other type of method to transfer annotations from characterized to uncharacterized proteins, or they construct, or there are a class of methods that can extract features that represent these nodes, that represent actually structure of these nodes across all of these networks, and these features can then be used for function prediction. So the first class, one of the type of method that belongs to the first class is GeneMania, and it's been state of the art for basically a long time. It's been published in 2008, and it kind of uses it integrates networks into a single network by using adaptive network weighting, and then it runs label propagation on top of this integrated network to transfer annotations from characterized to uncharacterized proteins. So the reason I mentioned this is that we're going to be using this method in our comparison study. So there are other types of methods that can be used for network integration, and there are more like network embedding methods, and network embedding is like a very recent approach. So the goal of network embedding is that you start with your network representing protein interactions, and that is represented as a GCC matrix, so n by n matrix. And the goal is to learn low-dimensional feature matrix n by k, so where we assign a k-dimensional vector to every protein in a network, where k is much, much smaller than n. So And the goal is that these features can preserve topological structure of the nodes in the network, meaning that basically proteins that are localized in the same part of the network or that are very similar are also localized in the same part of this low-dimensional feature space. And there is a bunch of these methods that are already proposed, such as node-to-vec, deep walk, line, etc. So the only problem with this is that these methods are all single network methods, and they cannot be applied to the situation where we want to integrate networks. Therefore, there was a recent approach that was proposed called mashup. It was published in 2016, and it actually is it's a type of matrix factorization-based method that creates these integrated or shared features by that are actually representing nodes across the set of networks. And then they use these features for all sorts of tasks, including function prediction. So this is a type of integrated network embedding methods, basically. And our method that I'm going to present in the next slide it actually belongs to this class. And we're also going to use this mashup method in our comparison study. Then I'm going to show you in a few slides. So our method it's called deep network fusion. Or DeepNF, and it's also pronounced deep enough, which you're going to see the reason why in a few slides. So it's a method that is based on a multimodal deep autoencoders, and we use multimodal deep autoencoder to learn these low-dimensional embeddings from a different network views, or as we call them, also modalities. Some of the conceptual advances of our method over mashup is that our method is a nonlinear dimensionality reduction method, which presumably should create features that are much better than just what you would obtain from linear methods, and so they are better in basically in learning features to describe these complex large-scale networks. Then the method can handle noisy links that are often present in some of these networks, and the reason is because it's based on autoencoders, and autoencoders are, at least in computer vision, very known for their denoising power, for example, in denoising images. And the third thing is that the method is efficient and scalable in a way that you can start with any number of networks you have, and you will end up with only a single feature matrix that will represent these nodes. So you can start with any number of n by n adjacency matrices representing your network, and you get to single n by k matrix where k is much, much smaller than n. And these feature vectors can be used for, in this case, for function prediction. So the method is also published, so the paper recently came out, so you can actually read about all the details that I'm not going to mention here, but there's also code that is freely available and can be used to extract features from any number of networks you have, or and then use these features for function prediction. So this is the core of our method, demonstrated on a single network. So in the next slide, I'll just show you how this works when we have multiple networks. So in this case, before using an autoencoder, we want to make features extracted representing nodes in the network, or proteins in this network, that are suitable for this deep learning technique here. So in order to do that, we run these two pre-processing steps. One is just a random walk that supposed to smooth profiles of each node in the network, right? If you just consider, for example, adjacency matrix, and you want to, for example, if you take one row in that adjacency matrix to represent your node, that's not that informative, because basically you only have direct neighbors encoded in that row in the adjacency matrix. Random walk is supposed to, the random walk is therefore basically taking into account the next neighbors and so on. Basically, it's supposed to create an adjacency, a matrix with probability profiles where probabilities represent basically how likely is that you're going to reach to that node from any other node in the network. So basically, this feature vector that you get after random walk is representing a node in the context in the context of all other nodes in the network. Then the next step is something that we kind of borrowed from natural language processing, which is construction of a positive pointwise mutual information matrix, which is just there to remove unreliable associations that we get from a co-occurrence matrix that we obtain after a random walk step. So, and after that, we use these like feature vectors to as an input to our autoencoder. So just note that none of these steps, so random walk plus PPMI, didn't change the dimensionality of our vectors. So if you started with n by n matrix, you are still left like without dimension. You'll see end up without dimension even after PPMI. So the reason, so and then the next step kind of is used to decrease the dimension. So the input to the autoencoder is this n-dimensional vector, and then we use these layers basically in the autoencoder to map this input vector to a low-dimensional space, eventually to reach to the bottleneck layer from which we extract these low-dimensional features. So let me give you just a feeling of the numbers, like so that you know, like basically in the yeast network that has like about 6,000 proteins, the input feature vector is 6,000, and you can just map that feature vector all the way down to, let's say, 600 dimensional feature vector. And then we use some of the visualization technique to represent this just in 2 dimensions, just to see the locations of these proteins. And so that if the approach works, then what you would expect is that basically the proteins that are localized in the same part of the network here, that are colored also depending on the type of function that they are performing, are also localized like you expect to be localized in the same part of the low-dimensional space. And then these features basically can be used for predicting function. So the way how we extend this approach when we have basically a collection of different networks is to use basically a multimodal deep autoencoder, or MDA. So this is basically the pipeline of our method. It's composed of these 3 steps. The first step is we run random walk on each individual network, and the second one is we construct PPMI matrices from each individual network, and then we use a multimodal autoencoder. So where in the first layer we handle each network separately, and we map them to a low-dimensional space, and then we concatenate these layers into a single layer, and then from there we can again apply some other layers until we reach to the bottleneck layer. So this first part of the autoencoder is called encoding part, and then in the second part we just reverse the process in which we just start with features and we reconstruct the data. So in this whole process, after we finish with the training of our autoencoder, we extract these compressed features from the bottleneck layer, and then we have a feature matrix that we can use for function prediction. So again, just to give you some numbers, so in our experiments, you're going to see in a few slides, is we start with 6 different networks. And for example, for yeast, it's about 6,000 by 6,000 matrices. So you have 6 matrices of 6,000 by 6,000. And basically, we first map them to 2,000 dimensional feature representation that is represented by this first set of hidden layers in the autoencoder. And after concatenating them, we map them further to low dimensional, so let's say to 2,000, and then to, let's say, 6,000 dimensional vectors to the bottleneck layer from which we extract these final features. And then after that, we can just train a classifier on these features and predict function. In our case, we just use an SVM. So this is the data we use. So we use basically these 6 networks that we obtain from StringDB, and I'm going to show you results separately for yeast and human. We also use 2 evaluation strategies. The first one is just adopted from the mashup paper, so it's identical as in the mashup paper. So it's a cross-validation because we wanted to make just a fair comparison with that method. So we just do the random splitting we have 80% of proteins that are in the training set and 20% of test proteins. On these 80% proteins, we run nested 5-fold cross-validation just to fit the hyperparameters of the SVM. And we report the results on these 20% of test proteins. And we do that on MIPS and gene ontology annotations that were also obtained from the mashup paper. And in the second evaluation strategy is more realistic and more Kappa-like, that we call temporal holdout. So we download annotations from 2015 and 2017 from UniProt Goa, and we divide the set of proteins in the following way. So we say the training proteins are those that didn't change, like their annotations did not change between 2015 and 2017. Then we call validation set are the proteins that had some annotations in 2015, but also gained new annotations in 2017. And we have a test set of proteins, are proteins basically that didn't have any annotations in 2015, but gained new annotations in 2017. So these GO terms that were gained by these test proteins are those that on which we're going to report our results. We also subset this set of GO terms by just looking at the GO terms that annotate between 10 and 1,000 of training proteins and that have more than 10 proteins in the validation set and 10 proteins in the test set. The reason is because we don't want to report the performance on very general GO terms. So here are some of the results. So this is the cross-validation separately on yeast, on MIPS terms, and that's on the left. On the right, you can see human results on gene ontology and specifically molecular function GO terms. So these different levels of MIPS represent different specificity of the terms. On the right, we split GO terms depending on how many, like on the number of annotations or the number of proteins they have. So we report the results on GO terms that annotate between 100 and 300 proteins. Then also on the proteins, and then also on the GO terms that annotate between 30 and 100 proteins, and also on the most specific ones which annotate between 10 and 30 proteins. So you can see here that we kind of outperform GeneMania and also Mashup it basically on every level, and below you can see just the top performing architectures. In the case of temporal holdout, you see even better results, and in which again we outperform and we get actually statistically significantly better results than Mashup, which is just a linear and a shallow technique for creating features. This is just to show you that basically that we get the best results only when we integrate all the networks together. So we get results that are better than any individual data source. And finally, I just want to show you some of the performance results on individual GO terms. So the performance is measured here by area under the precision recall curve for every GO term, and I'm plotting this against the also Mashup performance, and you see here that we kind of we got on the majority of GO terms, basically we outperform Mashup in the case of molecular function GO terms and cellular component, but also in the case of biological processes. Unfortunately, I don't have time to mention this. This is some of our future work. Right now, the DeepNF works on a single species level, but in case, let's say if you want to include more species, then we need some sort of mapping between these networks, and we kind of came up with a technique that is based on this global network alignment to map, let's say if you have yeast and human network, how do you construct universal features from this? And the reason for that is because basically we want to have more samples to train on and also more annotations. And also there are situations, for example, for some bacteria for which you don't have fully complete networks or interactomes, so therefore you can maybe use some other networks and plus this homology or sequence similarity basically, so with the help of sequence similarity basically to predict function, or to first create these features and then predict function. So, so this is just to conclude, basically we propose a method that is based on multimodal deep autoencoders that can handle multiple different types of networks and create features that are task independent, in sense that basically in the whole process of feature learning, we didn't use any of the functional labels. So basically the features that we create in this way are in some way universal. So it can be also used for other problems, but can also be used for, like, if you want to integrate them with other set of features, let's say structural data and so on, basically. And another thing is that the features are also general in a way that you can also use any other machine learning out-of-the-box machine learning classifier to, let's say, predict function. And we saw that basically the method, according to our results, outperforms both Mashup and GeneMania and both human and yeast, and in multiple levels of specificity of gene ontology and MIPS terms. So for all the details that I didn't mention here, you can come to my poster. It's today. Its poster number is 153, and I would just like to acknowledge the support from Simon's Foundation and also from my group, especially Meet and Emily, who are here, and they're also giving a poster presentation today, and these are the numbers of their posters. Thank you.
Thank you very much. We have time for a couple of questions. Back there, yes?
So I'm curious.
Can you come up to the microphone so that it will be recorded also?
So I was curious to see if you have measured centralities, for instance, of your networks, and see if those values are the ones that you're getting from your autoencoder, just to see if all that work is relevant, or it's as simple as calculating centralities, just a curiosity.
It's not simple as computing centralities, because
But have you?
instead of just
No, I know it's not as simple, but my question is if it is, so if since you're using an SVM, the question is if the attributes that you're using for training are much better than using centralities. Have you
no, we haven't made that kind of comparison study, but there are previous researchers that already confirmed that that's not that useful. The reason is, so you could actually manually engineer features for every node in the network by computing centrality, degree, clustering coefficient, and so on, and you can build this vector, right? But you're always going to end up with some limited information, because you're never going to be able to take all the information in the network. Like, for example, that we were able to compute by, I say, computing random walk. And plus, on the top of that, we have autoencoder that out of 6,000 dimensional feature vector kind of chooses the top 600, the most important features, right? So this feature learning and feature engineering, it's a completely different thing, and these techniques are actually much, much better than just manually computing these features and using that with some classifier, for example, to predict function.
Okay, so can I ask more questions? Just remember, back here at 4:40, we have a 40-minute coffee break now. Back here at 4:40. Remember, we have posters tonight, so I'll